So, we'll now simply practice a specimen paper from 2023 to strengthen our preparation and make sure we haven't missed anything from the ATP paper or the newer question styles. Remember, question patterns may change over time, but the concepts they test always come from the syllabus you've already covered. So, there's nothing to worry about. Let's begin. The first question is related to food tests. It asks, describe how you would use Benedict's solution to test for reducing sugar in some fruit juice. Include a suitable safety precaution in your answer. This is a direct food test question. We simply add Benedict's solution to the fruit juice and heat the mixture in a water bath. If a reducing sugar is present, the solution changes color. As a safety precaution, wear safety goggles and do not heat the test tube directly over a flame. So the answer we write is, add Benedict's solution to the fruit juice and heat the mixture in a water bath. If a reducing sugar is present, the solution changes color. As a safety precaution, wear safety goggles and do not heat the test tube directly over a flame. The next question asks, the students also decided to test the distilled water with Benedict's solution. Write the result of this test in table 1.1. Since distilled water contains no reducing sugar, Benedict's solution will show no color change and remain blue. So the answer we write is, blue. The next question asks, explain why the students tested the distilled water. Distilled water is used as a control. It shows the expected result when no reducing sugar is present, allowing the students to compare it with the glucose solutions. So the answer we write is, the distilled water was used as a control to show the color of Benedict's solution when no reducing sugar is present. The next question asks, state 3 other variables that the students should have controlled to make their results comparable. Whenever you carry out a fair test, all variables except the one being investigated should remain the same. So the answers we write are, volume of the glucose solution, volume or concentration of Benedict's solution, and temperature of the water bath. The next question asks, using the results in table 1.1, suggest what you can conclude about the percentage concentration of glucose in solution X. Explain how you reached this conclusion. The color obtained is yellowish-orange, which lies between the yellow color for 0.3% glucose and the orange color for 0.4% glucose. So the answer we write is, conclusion is the glucose concentration is between 0.3% and 0.4%. And the explanation is, the solution was yellowish-orange, which is an intermediate color between the yellow result for 0.3% and the orange result for 0.4%. The next question asks, the students found it difficult to decide the exact color of solution X after testing it with Benedict's solution. Suggest a reason for this difficulty and a method which could be used to improve their confidence in deciding the color. Sometimes color judgment is subjective, meaning different people may describe the same color differently. A color chart provides a standard reference and improves accuracy. So the answer we write is, difficulty. Color interpretation is subjective, so different people may describe the color differently. For improvement we will write, compare the solution with a color chart or have the same person record all colors. The next question asks, describe how the students could use a 1.0% glucose solution to produce 5 cubic centimeters of a 0.5% glucose solution. Since the required concentration is half of the original concentration, the solution should be diluted by mixing equal volumes of glucose solution and distilled water. So the answer we write is, measure 2.5 cubic centimeters of the 1.0% glucose solution and add 2.5 cubic centimeters of distilled water to make a total volume of 5 cubic centimeters of 0.5% glucose solution. The final question asks, suggest how the students could separate any solid from a solution and obtain its mass. To separate the solid, the mixture should first be filtered. The solid collected on the filter paper is then dried before its mass is measured using a balance. So the answer we write is, filter the mixture to separate the solid from the solution. Dry the solid and then measure its mass using a balance. Now let's move on to question 2. Now this is an analytical question based on data handling. It gives you a piece of information about the carrots and says, carrots are a food containing vitamin C. They can be frozen to be preserved and stored. Some students measured the vitamin C content of fresh and frozen carrots. They then boiled the carrots in water and measured the vitamin C content again. Their results are shown in table 2.1. Here you can see a table now. The first question asks us to construct a bar chart of the data in table 2.1 on the grid. So here's how you have to draw the graph. Remember that whenever you are asked to draw a bar chart, first draw and label both axes. Here, in this case, on the horizontal axis, write the types of carrots, and on the vertical axis, write vitamin C in milligrams per 100 grams. Next, choose a linear scale that uses at least half of the grid. Now plot 4 equal width bars with heights of 5.9, 3.6, 2.5, and 2.3. Remember to leave equal spaces between the bars because these are separate categories and the bars must not touch. The next question asks, state which boiled carrots contain the most vitamin C. For this, simply compare the two boiled carrot values in the table. Fresh boiled carrots contain 3.6 mg per 100 grams, while frozen boiled carrots contain 2.3 mg per 100 grams. Therefore, fresh boiled carrots contain more vitamin C. So the answer we write is, fresh boiled carrots. The next question asks, suggest two conclusions the students could reach from these results. So by looking at the table, you can clearly see that boiling reduces the vitamin C content because both fresh and frozen carrots contain less vitamin C after boiling. You can also observe that freezing reduces the vitamin C content because frozen carrots contain less vitamin C than fresh carrots. So the answers we write are, boiling decreases the vitamin C content of carrots, and freezing decreases the vitamin C content of carrots. The final question asks, carrots can be cooked by heating them in an oven or by boiling them in water. Describe how you would do this investigation. Whenever you describe an investigation, begin by making it a fair test. We will use carrots of the same type, or pieces from the same carrot, and cut them into equal masses. Before cooking, we will measure the vitamin C content using the vitamin C test. Next, we will cook one group by boiling and the other group in an oven, making sure both groups are cooked for the same amount of time and at the same temperature. After cooking, we will measure the vitamin C content again using the same vitamin C test. Finally, we will repeat the investigation several times and compare the results to make them more reliable. So the answer we write is, use carrots of the same type or the same carrot and cut them into pieces of the same mass. Measure the vitamin C content before cooking using the vitamin C test. Cook one group by boiling and another group in an oven, using the same temperature and the same cooking time for both methods. After cooking, measure the vitamin C content again using the vitamin C test. Repeat the investigation several times and compare the results to improve reliability. Now let's look at the final question. It says, the potato is a plant that can store starch grains in its cells. Figure 3.1 shows some of these starch grains as seen under a microscope. The figure shows several starch grains inside potato cells, labeled D, E, F, and G. The first question asks, identify the structure labeled D. If you look carefully at the figure, label D points to the outer boundary of the cell, which is the cell wall. So the answer we write is, cell wall. The next question asks, draw the starch grains labeled E, F and G as they appear in figure 3.1. For this question, we need to follow the drawing instructions carefully. First, draw only the starch grains labeled E, F and G, and do not include anything else from the figure. Grain F must be at least 60 mm long, so before drawing, use a ruler to measure a length of 60 mm for accuracy and then draw grain F larger than 60 mm along with grains E and G beside it exactly as shown in the figure above. You also have to make sure that your drawing covers 70% of the given space, so make it accordingly. Also, make sure the grains are touching each other, keep their relative sizes and shapes correct, and use clear, single continuous lines only, without any sketching or shading. The next question asks, on your drawing, draw a line to indicate the maximum length of grain F. Measure this length and record it. Now when your drawing is complete, draw a straight line from one end of grain F to the other to show its maximum length you have actually drawn. Then use a ruler to measure that line and write the measurement in millimeters beside it. My measurement in this drawing is 120 millimeters, so this will be my image size. Then, the next question asks, the actual length of grain F is 0.03 millimeters. Calculate the magnification of your drawing to the nearest whole number. Now, to calculate magnification, use the formula. Magnification equals the length of the drawing divided by the actual length. So, our length was 120 mm, which is image size, and we will divide it by 0.03 mm, which is our actual size, giving a magnification of 4,000 times. Remember that we always use this small x symbol to represent magnification. Next, the final question asks, describe how to prepare a slide of potato tissue to observe starch grains as clearly as possible under a microscope. Now recall, to prepare the slide, first cut a very thin section, or gently scrape a small amount of potato tissue, and place it onto a microscope slide. Next, add a drop of iodine solution to stain the starch grains. Then carefully lower a cover slip over the specimen, making sure to avoid trapping air bubbles. Finally, wipe away any excess iodine solution before placing the slide under the microscope for observation. So the answer we will simply write is, cut a thin section or scrape a small amount of potato tissue and place it on a microscope slide. Add a drop of iodine solution to stain the starch grains. Carefully lower a cover slip, avoiding or removing air bubbles, and wipe away any excess stain before observing under the microscope. With that, we come to the end of our complete O-Level Biology 5090 course. Thank you so much for watching, goodbye, and take care.